In the early 1960s, a sleeping pill sold in over 40 countries caused thousands of children to be born with missing or severely shortened limbs. The drug was pulled from the market, and for 50 years, nobody could explain exactly why it worked or why it had done that.
The gene is CRBN, cereblon, chromosome 3, band P26.3. It encodes a protein that acts as the selection door of the cell's protein destruction machine, an E3 ubiquitin ligase. When cereblon chooses a protein, it tags it for disposal. The question was, what makes it choose?
In Tokyo, biochemist Hiroshi Honda had spent decades perfecting a new way to find what drugs actually bind. His team coated magnetic nanoparticles, FG beads, with thalidomide, then dipped them into human cell extracts. 2 proteins stuck. One of them was cereblon. Honda and Ito published in Science in 2010. 50 years of mystery solved in a pulled-down experiment.
What made FG beads work where earlier attempts had failed was surface chemistry. Thalidomide is hydrophobic. Older agarose matrices stuck to everything, drowning the true signal in noise. FG beads are magnetic, non-porous, and low background. Coat the bead with thalidomide, hold a magnet to the tube, and you get back your drug, and whatever was bound to it.
Cereblon is the gatekeeper of CRL4, a molecular machine that tags proteins with ubiquitin and feeds them to the proteasome. Normally, cereblon picks specific proteins for destruction, but when lenalidomide binds its tri-trip pocket, the drug acts as molecular glue, creating a new surface that recruits neo-substrates, proteins CRBN would never touch on its own. For myeloma, those neo-substrates are Icarus and Aiolos, transcription factors the cancer cells depend on to survive. The drug doesn't block them, it sentences them to death.
Multiple myeloma is a cancer of plasma cells. No cure. 2 groups showed in 2014 that lenalidomide works by degrading Icaros and Aiolos via CRL4-CRBN. Before these drugs, median survival was around 3 years. Lenalidomide became one of the best-selling cancer drugs in history. The worst pharmaceutical story of the 20th century had quietly become one of its best.
Once scientists understood how cereblon worked, the question followed, could you hijack it deliberately? Build a molecule with 2 arms. One end grabs cereblon, the other grabs any protein you want destroyed, and you have a machine that can eliminate almost any target in the cell. These are PROTACs, proteolysis targeting chimeras. The first CRBN-based PROTAC was designed in 2015 to destroy BRD4. In 2026, the first PROTAC ever approved by the FDA was a CRBN recruiter, vepdegestrant, for estrogen receptor-positive breast cancer. Thalidomide, the drug that was wrong twice, turned out to be a master key.
1954, a sedative. 1961, catastrophe. 2010, cereblon discovered. 2015, PROTACs begin. 2026, the first PROTAC approved, the gene channel.